Hello everyone, myself Professor Khemchen Surana. So today we will discuss the last and the important class of the anti-cancer agent, that is what miscellaneous anti-cancer agents. In that we will see the different miscellaneous drugs which will act as a anti-cancer or the which will act against the cancer. So in that initially we will see the protein kinase inhibitors. Okay, so there will be a short note will be asked in examination about your the tyrosine kinase inhibitors or protein kinase inhibitors. So the tyrosine kinase inhibitors examples from this category is imatinib, jeftinib, erlotinib and sorafenib, also the sumitinib and lapatinib. So these are all what the tyrosine kinase inhibitors, means protein kinase inhibitors. At the end there is what nib, okay, tinib or trinib. So these are all what tyrosine kinase inhibitors. So imatinib is what a first selectively targeted drug to be introduced inhibit a specific tyrosine kinase protein. Okay, so basically these drugs, okay, whatever the tyrosine kinase, okay, the tyrosine kinase is the enzyme which will involve into the development of cancers, okay, different kinds of cancers. So these drugs were used to inhibit these enzyme. Okay, so this is what the first targeted drug which will inhibit the specific tyrosine protein kinase, that is what BCR-ABL tyrosine kinase, which is expressed by CML cells. Okay, and related receptors tyrosine kinases includes the PDGF receptor that is constitutively active in the dermatofibrosarcoma, protuberans, stem cell receptors and c-kit receptors active in GIST. Okay, so whatever these drugs, okay, which will inhibit the tyrosine kinase, okay, specifically BCR-ABL, okay, which is expressed by these CML cells, and also the different kinds of receptors like PDGF, c-kit receptors and stem cell receptors. Okay, and useful in case of dermatofibrosarcoma, protuberans. Okay, so this is what about the protein kinase inhibitors. Here the different structures of protein kinase inhibitors given. So specific BCR-ABL kinase inhibitors given over here. Okay, so in that example imatinib mesylate, okay, nilotinib hydrochloride and dasatinib. So you can see there is what pyrimidine ring is what the basic thing and quite complex structure here. Okay, just practice these structures. Okay, this is about what BCR-ABL kinase inhibitors. Okay, 3 examples. Now next is what EGFR kinase inhibitors. Okay, in that example is erlotinib hydrochloride and jeftinib. Okay, so just need at least you need to remember at least 2 structures, okay, from every class of these kinase inhibitors. Okay, so this is what about the structures. Okay, just practice all these structures. Okay, these are important as per your examination. Then next class from this protein kinase is what EGFR and HER2 kinase inhibitors. Okay, so in that example is lapatinib distosylate. Okay, so this is the structure. Okay, again pyrimidine kind of ring system. Okay, then furan will be there, NH. Okay, then further sulfonate group will be there. Okay, so this is what dietosylate. Okay, because of this sulfate group. Okay, so next class is what VEGFR kinase inhibitors. In that example is pazopanib hydrochloride. Okay, so pazopanib hydrochloride structure is there. Okay, you can see the basic pyrazole ring system. Okay, imidazole and pyrazole. Okay, imidazole 1,3 nitrogen, pyrazole 1,2 nitrogen. Okay, in case of pazapamib. Then sumitinib. Okay, indole kind of oxyindole kind of a ring system. Okay, basic with pyrrole. Okay, then next drug is what sorafenib tosylate. Okay, so these are what the different protein kinase inhibitors. Just remember the at least one structure from every class and different examples. Okay, so this is about what protein kinase inhibitors. So these protein kinase inhibitors very successful in case of what phase of CML, okay, and metastatic c-KIT GIST, okay, also indicated in what dermatofibrosarcoma protuberans. So useful in this case is what dermatofibrosarcoma protuberans, okay. So resistance develop mainly due to the point mutation in BCR-ABL tyrosine kinase, okay. So whatever these BCR-ABL tyrosine kinase, it is what uh, enzyme which will grow in case of the cancerous condition, okay. So some kind of resistance were developed, okay, because of mutation, okay. So metabolized by mainly CYP3A4, okay, and metabolites excreted in feces through the bile. So metabolism is there, so through CYP3A4 enzyme these were metabolites, and excretion through what feces through bile, okay. So adverse drug reaction of these protein kinase inhibitors is basically abdominal pain, okay, vomiting, fluid retention, periorbital edema, pleural infusion, myalgia, and CHF, okay, chronic heart failure. So this is what about the ADR. So dosatinib and nilotinib are similar drugs used in the case of imatinib resistance. If the imatinib resistance were causes, so in that case the dosatinib and nilotinib, these drugs were useful. So this is about what the protein kinase inhibitors. Now next miscellaneous class is what enzymes, okay. So enzymes, they were act as an anti-cancer agent. So from example is what L-asparginase and asparginase. These are what the examples of the enzymes, okay, useful as an anti-cancer. So L-asparginase basically an enzyme, okay, which is isolated from the E. coli, okay, from E. coli this will what isolated, and these enzymes basically causes the catabolic depletion of, okay. So these were causes what catabolic depletion of serum aspargine to aspartic acid, okay, means whatever the aspargine were converted into what aspartic acid by using this L-asparginase enzyme, and ammonia resulting in reduce blood glutamine level and inhibition of protein synthesis. Once this conversion has been occur, okay, by using this L-asparginase, okay, so this aspartic acid into the cell, again it is what converted into what your aspargine, and this will helpful for what your protein synthesis, okay. So neoplastic cells require external source of aspargine, okay. Whatever the neoplastic cells or the cancerous cells, okay, these cells having what low level of this asparginase, okay. So these cells, they don't have too much or the proper or the required concentration of these enzymes. So if the concentration of these enzymes decreases, the protein synthesis were decreases and the cancerous cells were inhibited. So this will treat the childhood acute leukemia, okay, and causes the anaphylactic shock. So this is what about the L-aspargine. So here the mechanism of action of L-aspargine has been discussed, okay. So into the blood basically, so these enzymes were useful in case of what leukemia, okay. So in case of a normal cell and in case of neoplastic cells, here two diagrams given. So the first diagram is what in case of normal cells. So the aspargine basically converted into what aspartate by using this exogenous asparginase enzyme, okay. Now further this aspartate were enters into what normal cell, okay, and here this aspartate again converted into the aspargine by using aspargine synthetase enzyme by the use of what glutamine, which will convert it into glutamate, okay. So now further whatever this conversion of aspartate to aspargine, okay, into the normal cell, this aspargine will helpful or the useful for the protein synthesis and the growth of new cells. The normal cells having a high level of aspargine synthetase and are able to make sufficient aspargine for protein synthesis and cell growth. So as we know, the normal cell is having what this asparagine enzyme having in proper concentration. That's why it will help to protein synthesis. But in case of what, neoplastic cells, you can see the second diagram. Okay, so the asparagine will converted into what, aspartate by exogenous asparaginase enzyme. Now this aspartate where enters into the neoplastic cells, and here the neoplastic cells having what, the low level of asparagine synthesis, and they were unable to make sufficient asparagine for protein synthesis and cell growth. Okay, so whatever the neoplastic cells having a low level of this asparagine synthesis, that's why whatever the aspartate were not converted into the asparagine by using glutamine and glutamate, therefore the protein synthesis has been inhibited. So this is what the mode of action of this L-asparaginase. Okay, now the next example from this miscellaneous class is hydroxyurea. Okay, the structure of hydroxyurea is given over here. You can see NH2 C double bond O NHOH. Okay, so hydroxyurea is the analog of urea and which inhibit the ribonucleotide reductase enzyme. Okay, resulting in the depletion of deoxynucleoside triphosphate pools. Okay, thereby inhibit the DNA synthesis. Okay, so this is what the inhibitor of ribonucleotide reductase, and once this ribonucleotide reductase has been inhibited, okay, there is what, depletion means decrease of deoxyribonucleotide triphosphate pool, means whatever the library of deoxynucleotide triphosphate has been depleted. Okay, therefore which inhibited the DNA synthesis. So this hydroxyurea specifically S phase specific unit. Okay, and useful in the treatment of melanoma and chronic myelogenous leukemia. Okay, so here the mode of action given, the ribonucleotide were converted into deoxynucleotide. This deoxynucleotide is useful for what, DNA synthesis. Okay, with the this conversion of ribonucleotide by ribonucleotide to deoxynucleotide by using this ribonucleotide diphosphate reductase, and this hydroxyurea will inhibited this ribonucleotide diphosphate reductase enzyme. So therefore the conversion of ribonucleotide to deoxyribonucleotide has been inhibited and DNA synthesis also inhibited. So adverse effect of this hydroxyurea is myelosuppression. Okay, hypersensitivity, hyperglycemia, hypoalbuminemia. Okay, so these are what, different adverse effect. So next example of this miscellaneous class is mitoxantrone. Okay, so mitoxantrone structure has been given over here. So structure is resembles or similar like what your anthracycline. Okay, so anthracycline ring will be there and NHCH2 twice, NHCH2 twice OH at both end of the first ring. Okay, so these mitoxantrone binds to the DNA to produce strand breaking. Okay, so like anthracyclines, okay, they were binds to the DNA and produces this strand breakage. Okay, and by this inhibited the DNA as well as RNA synthesis. And these mitoxantrone useful in the treatment of and useful for the treatment of pediatric as well as adult acute myelogenous leukemia, non-Hodgkin's lymphomas and breast cancer. So in these cases these mitoxantrone has been useful, and these were causes the cardiac toxicity. This is what the side effect of mitoxantrone. Just draw the structure and practice it. So now the next therapy is what, from the immunotherapy. Okay, we have to discuss here. Okay, so here the non-specific cancer immunotherapies and their adjuncts. Okay, so this may ask in examination for your 3 marks. Okay, and important as per your examination. So the non-specific immunotherapies don't target cancer cells specifically. Okay, so these non-specific immunotherapy, they were not target to what, your cancer cells, but they will stimulate the immune system in more general way, but this can still sometimes leads to a better immune response against cancerous cell. Okay, so they were not targeting the cancerous cell, but they were increases the immunity. Okay, So some non-specific immunotherapies are given by themselves as a cancer treatment. Okay, so other are used as the adjunct, okay, along with their main treatment to boost the immune system to improve how well another types of immunotherapy such as vaccine, okay, they will work. Okay, means the some kinds of non immunotherapy explained over here, but the other kind of non-specific immunotherapies, okay, they were also explained, but they were not as a proper non-specific immunotherapy, okay, but they were called as, we can call as a adjuvant, okay, means what, to boost the system, okay, to improve their immunotherapy, okay, such as the vaccines is what example. So some are used by themselves against some cancer and adjuvant against the others. Okay, so here the cytokinin is what examples, okay, from these non-specific cancer immunotherapy. Okay, so cytokinins are the chemicals made by some immune system cells. Okay, they are crucial in controlling the growth and activity of other immune system and cells as well as blood cells. Okay, so these are what the useful to make the immune system active, okay, and controlling the growth as well as activity of immune system into cell as well as blood cells. So cytokinins are injected either under the skin or into the muscles or into the veins. Okay, the most common one is discussed over here. So example of these cytokinin is what interleukins given over here. So interleukins are the group of cytokinins, okay, that act as a chemical signal between white blood cells. Okay, so these are what the between these white blood cells, WBCs, these are what chemical signals. Okay, so interleukin 2, that is what IL-2 we can call it, helps the immune system cells to grow and divide more quickly. Okay, so these interleukins were what help to what grow as well as divide quickly. A man-made version of the interleukin 2 is approved to treat advanced kidney as well as metastatic melanoma. So these interleukins 2 were useful in case of was kidney cancer as well as metastatic melanoma. So interleukin 2 can be used as a single drug treatment for this cancer, okay, or it can be combined with chemotherapy or with other cytokines such as interferon alpha. Okay, using interleukin 2 with this treatment might help make other more effective against some cancers, but side effects of this combined treatment are also increases. Okay, so whatever these interleukins as well as the interferons, they were also useful in what combination or the combination with the other treatment, they will increases the effect, but also they will increases their side effects. Okay, so this is about what your interleukins. Now the side effect of these interleukins can be includes basically flu like symptoms as will be there, so such as chills, fever, fatigues and confusion. So these are what the side effect of interleukin 2. So most of the peoples gain weight. Okay, so since some peoples the weight were gained, and some are having nausea, vomiting as well as diarrhea. Many people develops low pressure which can be treated other medicines. Rare but potentially serious side effects includes an abnormal heartbeat, chest pain and other heart problems. So because of these possible side effects, if interleukins 2 is given in high doses, it must be done in hospital. Okay, so whatever these, because of the side effects, so these interleukin were given into the high doses and must be done in hospital, means this treatment should be carried out in hospital only. Okay, the other interleukins such as interleukin 7, interleukin 12, interleukin 21 are now being studied for the use against the cancer, both as an adjunct as well as standalone agent. Okay, so next is what interferons. Okay, so the interferons short notes were asked in examination. So interferons are also what the chemical agents or the chemicals that helps the body to resist the virus infections as well as cancers. Okay, the types of interferons we can call in short IFN, okay, are named after three letters of the Greek alphabet. So there are three types of these interferons: interferon alpha, interferon beta, and interferon gamma. So from these all three, the interferon alpha is used to treat the cancer. Okay, so only interferon alpha is used to treat the cancer. It boosts the ability of certain immune cells to attack cancerous cells. It may also slow the growth of cancerous cells directly, as well as blood vessels that tumors need to be grow. Okay, so about of these three interferon types, alpha, beta, and gamma, the alpha has been useful against your cancer and used to boost the your immune cells and attack to the cancerous cells. Okay, so interferons basically used to treat the different types of cancer such as hairy cell leukemia, chronic myelogenous leukemia, you can call CML in short, okay, follicular non-Hodgkin lymphoma, cutaneous skin T cells lymphoma, kidney cancers, melanoma, Kaposi's sarcoma. So in these different kinds of cancers, these interferon were useful. Now here the side effects of some interferon has been given. So the flu-like symptoms like chills, fever, headache, fatigue, loss of appetite, nausea, vomiting. Okay, these are what the flu-like symptoms of interferons. Now also the slow white blood cells count. Okay, so whatever WBC counts has been low. Okay, so low white blood cells count will be there, which increases the risk of infections, skin rashes, and also thinning the hairs. Okay, so these are what the different side effects of interferons of your like flu like symptoms is now low WBC counts, skin rashes, and thinning hairs. So these are different kinds of your side effects of interferons. Now here the miscellaneous class and your anti-cancer agent chapter, that is what the broad chapter, or we can call the important as well as the big chapter, has been finished over here. Okay, now in next session we will start the new chapter, that is what antibiotic. Till that, thank you and happy learning.